Okay, next subtopic in polymerase chain reaction is its principle. So we'll study what is principle of PCR. Polymerase chain reaction involves many rounds of DNA synthesis. All DNA synthesis reactions require a template. The template means the sequence of DNA which needs to be copied, a primer, a small segment of DNA which bind with this template, then in this enzyme DNA polymerase, which actually adds new nucleotides in the growing primer, in the growing DNA segment, and then a supply of nucleotides. In standard PCR, generally two primers are used: forward primer and reverse primer. The primers flank the target sequence in a DNA for its amplification, and in this way it starts forming new copy of the desired gene. This DNA synthesis therefore proceeds across the region between the primers. Each round of DNA synthesis is known as a cycle and involves basically three reactions. And what are those three reactions? Number one is denaturation, number two is primer annealing, and number three is primer extension. Denaturation, as the name indicates, in this process, double-stranded DNA, which is to be copied, or the part of which is to be copied, is heated, and in this way it is changed into two single-stranded DNA segments. And each strand is then copied. Primer annealing, the part is the process in which primer that combine with a specific sequence on the parent DNA molecule. And then primer extension, new nucleotides which are added, they are added by DNA polymerase, and in this way the new DNA molecule is synthesized and it is extended, or its extension takes place. So these three basic steps are involved in this PCR. The products of each synthesis reaction can be used as templates for further DNA synthesis. So हमें एक कॉपी से जो दो कॉपी मिलती हैं, वो जो दो कॉपियां होती हैं, then they act as template for the synthesis of new DNA molecule. And in this way, 4 molecules are formed. These 4 templates, they act as source for the formation of new DNA molecules, and so on. This results in exponential amplification of the target sequence in a very limited time. In a matter of few hours, we get billions of copies of the desired gene.